Hello student, good morning. Myself Sri K. D. Borse sir, biology subject teacher. In previous lecture we learned cell structure and organization chapter content concept of cell. We learn basic information about microscope because cell see clearly need microscope, so we learn some basic information about microscope. Cell size in plant and animal, also we learn postulate of modern cell theory and concept of totipotency. Today we are going to learn some more concept including cell structure and organization chapter, kinds of cell. All living organism grouped into two categories, prokaryote and eukaryote. Prokaryote means those organisms made by prokaryotic cell, those organisms made by eukaryotic cell. We learn prokaryotic cell in today's lecture. What is prokaryotic cell? What are the characteristics of prokaryotic cell? Cell envelopes of prokaryotic cell and cell organelles of prokaryotic cell. So let us start today's first concept, kinds of cell. All living organisms grouped under two categories, prokaryotes and eukaryotes. What is different between prokaryotes and eukaryote? Prokaryote means those organisms made by prokaryotic cells, such organism called as prokaryote, and those organisms made by eukaryotic cells, such organism called as eukaryote. So this is the difference between prokaryote and eukaryote. Best example, prokaryote, bacteria; eukaryote, plant, animals, eukaryotes. Cells are also two types on the basis of prokaryote and eukaryote. Prokaryotic cell and eukaryotic cell. Prokaryotic cell made by prokaryote, eukaryotic cell made by eukaryote. So on the basis of these two category of organisms, cells are also two types, prokaryotic cell and eukaryotic cell. We learn one by one prokaryotic cell and eukaryotic cell. In today's lecture, we discuss prokaryotic cell. What is prokaryotic cell? Prokaryotic cell is one of the type of cell in which well-organized nucleus is absent. Such cell called as prokaryotic cell. It is one of the type of cell in which well-organized nucleus is absent. Such cell called as prokaryotic cell. ज्या सेल मध्ये वेल ऑर्गनाइज्ड न्यूक्लियस ॲबसेंट असते ती सेल प्रोकॅरियोटिक सेल असते. इन मराठी वी लर्न आदि केंद्र की पेशी. Example, bacteria cell is prokaryotic cell. Bacterial cell is prokaryotic cell. Most important characteristics of prokaryotic cell: well organized or well defined nucleus is absent. Well organized or well defined nucleus is absent, and membrane bound cell organelles are also absent. Always remember these two characters are most important about prokaryotic cell. Well organized nucleus or well defined nucleus is absent. Also membrane bound cell organelles are absent. What are the membrane bound cell organelles? Like mitochondria, Golgi complex, endoplasmic reticulum, vacuoles are membrane bound cell organelles. Such cell organelles are absent in prokaryote. So this is the two most important characteristics of prokaryotic cell. We learn structure of prokaryotic cell. Prokaryotic cell has chemically complex protective covering called as envelopes. Envelopes means protective covering, and in prokaryotic cell these are chemically very complex. Such envelopes are three layered structure show in prokaryotic cell. Such envelopes are three layered seen in prokaryotic cell. First one is glycocalyx, second one is cell wall, and third one is plasma membrane. How many envelopes are seen in prokaryotic cell? Three. Which are those? Glycocalyx, cell wall, plasma membrane. Glycocalyx is outermost envelope or protective covering of prokaryotic cell, while cell wall is middle below the glycocalyx, protective covering or envelope of prokaryotic cell, while plasma membrane is innermost protective covering of prokaryotic cell. This is the prokaryotic cell. Here this one is outermost covering of prokaryotic cell called as glycocalyx. Labeled them. This is the glycocalyx. Clearly seen, glycocalyx. Here cell wall below the glycocalyx. This one is cell wall. Labeled them. This is the cell wall. Seen here, cell wall is a middle layer or second envelope below the glycocalyx. This one is cell wall, very thick layer, and this one is plasma membrane. Seen here, plasma membrane is innermost envelope of prokaryotic cell. Here noted, glycocalyx is outermost, cell wall is middle, and plasma membrane is inner envelope seen in prokaryotic cell. Means in prokaryotic cell there are three envelopes: glycocalyx, cell wall, plasma membrane. We'll learn one by one in detail. First one is glycocalyx. What is glycocalyx? It is outer covering or envelope of prokaryotic cell. It is slim capsular layer because it look like capsule covering of protective nature provide to prokaryotic cell. Slim capsular layer glycocalyx. Function: glycocalyx envelope help adhesion of bacterial cell. Means adhesion means fixation nature. Due to adhesion nature or fixation nature, bacteria fix to any substratum. show me here diagram. This is the glycocalyx, is outermost envelope. Adhesion nature due to this bacterial cell is fixed to substratum. Now second envelope of prokaryotic cell called as cell wall. Cell wall is present below the glycocalyx. It is made up of peptidoglycan, such bacteria called as gram-positive bacteria. If cell wall is made up of murine material, such bacteria called as gram-negative bacteria. Means on the basis of cell wall, bacteria consist of two types. Its cell wall is made up of peptidoglycan material. Such bacteria is gram-positive bacteria, while the cell wall of bacteria is made up of murine material. Such bacteria called as gram-negative bacteria. Function of cell wall: it gives mechanical support to the cell. Function of cell wall: it gives mechanical support to the cell. Due to cell wall shape maintain of prokaryotic cell provide mechanical support is also to the cell. Now third envelope called as plasma membrane, also called as cell membrane. Plasma membrane also called as cell membrane. It made up of bilayer phospholipid. Always remember, plasma membrane is bilayer made up of phospholipid. Function: help intercellular transport. This is the plasma membrane, is actual cytoplasmic covering. This plasma membrane is specially for intercellular transport, means inside the cell content material transfer from inner to outside, while outside material transfer from outside to in the cytoplasm. So this is the main function of transport inner cellular material with the help of plasma. To this we detail discuss all three envelopes of prokaryotic cell, like glycocalyx, specially for adhesion to the bacterial cell. Cell wall gives mechanical support to the cell. Plasma membrane help intercellular communication, means each envelope provide special or different function to the prokaryotic cell or bacteria, but a common main function is provide protection to the prokaryotic cell or bacteria. Hope that you understood everything. Now we learn some structural point of view. Prokaryotic cell. Number 1, prokaryotic cell of motile bacteria, means movable bacteria, show either flagella or cilia. With the help of flagella or cilia, help rotatory movement to the bacteria, means both organs like flagella and cilia help rotatory movement to the bacteria. Seen here diagrammatic representation. This is the flagella. Due to this flagella, rotatory movement takes place, bacteria. So this bacteria called as motile bacteria or motile prokaryotic cell. Second point, this is the flagella. Some other surface show projection. Some other surface show projections are called as pili and fimbri, seen here. Some other surface show projections. Such projection called as pili or fimbri, labeled them pili or fimbri. These are the pili or fimbri. Such pili which help intercellular communication, and fimbri which help cleaning and support. Here noted, pili and fimbri are most important function provide to the prokaryotic cell or bacteria. Pili help intercellular communication, while fimbri help cleaning and support. This is the pili present on outer surface of the bacteria. Now point number 3, the cell membrane show infoldings called as mesosome. Cell membrane show infoldings called as mesosome. Function of mesosome: help cell wall formation, also help in DNA replication. Senior diagrammatic representation. What is actual mesosome? This mesosome is attached to plasma membrane. You know, plasma membrane is innermost layer. Such plasma membrane attached with mesosome. Such mesosome provide two functions: help in cell wall formation, also help in DNA replication. मेसोझोम सेल वॉल फॉर्मेशनचं काम करतो. सेकंड, DNA रेप्लिकेशन प्रोसेसला मदत करतो. You know, DNA replication means DNA duplication or Xerox of the DNA. Such process help mesosome. So this is the two functions of mesosome. Point number 4. Some bacteria, example photosynthetic cyanobacteria, show chromatophores. Function of chromatophore: carry photosynthetic pigment. Point number 5. The cytoplasm contain dense particles called as ribosomes. Seen here, diagrammatic representation. These are the dense particles, black colored, seen in the cytoplasm, called as ribosome. So these dense particles called as ribosome present in the cytoplasm of the prokaryotic cell. In prokaryotic cell or bacterial cell, ribosomes are 70S type. Most of time this question is noted: which type of ribosome present in bacterial cell? 70S type. Such 70S type bacteria composed of its larger unit is 50S type, while smaller unit is 30S type. Means larger unit 50S type and smaller unit 30S type are combined to form 70S type ribosome present in bacteria. Such ribosome is most important for help in protein synthesis because ribosome is a protein synthesis factory. So ribosome help protein synthesis process. Up to this we detail discuss. Here noted 5 different structural basic points of prokaryotic cell. Hope that you understood everything. Now point number 6. Cytoplasm of prokaryotic cell contain nucleoid or chromosome and plasmids. Here noted this part is nucleoid or chromosome present in the prokaryotic cell, while this is the plasmid of prokaryotic cell. We learn one by one. First one is nucleoid or chromosome. Nucleoid or chromosome, it remains attached to mesosome, here clearly seen. This one is mesosome. This mesosome is actually attached to plasma membrane and mesosome with attached nucleoid. Nucleoid or chromosome of prokaryotic cell are also called as genetic material of bacteria because this nucleoid or chromosome is transferred from one generation to next generation. So this is called as genetic material of bacteria. Such genetic material of bacteria consists of single chromosome made up of circular or coiled DNA. This one is actual circular or coiled DNA of prokaryotic cell. So this is also called as genetic material of bacteria. Such circular DNA show very typical type of replication called as theta model of replication. Which type of typical replication seen in prokaryotic cell genetic material or circular DNA or coiled DNA? Theta model of replication. This DNA not associated with histone protein like eukaryote. Always remember, in eukaryotic DNA associated with histone protein, but prokaryotic DNA not associated with histone protein. Up to this we detail discuss. Nucleoid or chromosome is a genetic material of bacteria. Now we learn cytoplasm contain plasmid is also most important because in prokaryotic cell show one or two additional molecule of circular DNA. In prokaryotic cell show one or two additional molecule of circular DNA, also called as mini chromosome, and this structure called as plasmid. What is plasmid? In prokaryotic cell show one or two additional molecule of circular DNA, also called as mini chromosome. Such structure called as plasmid. Seen here diagrammatic representation. Plasmid seen in prokaryotic cell. No doubt plasmid is also DNA, but it is not a nucleoid DNA. It is a additional molecule of circular DNA, also called as mini chromosome. Plasmids are actual extra chromosomal DNA. Plasmids self-replicating DNA means it is also transfer from one generation to next generation, so it has also nature self-replicating. Plasmids are of two types, F plasmid and R plasmid. F plasmid and R plasmid. F plasmid specially for reproduction, while R plasmid for resistant against antibiotics. Means it is most important for prokaryotic cell because R type plasmid provide resistant powers against antibiotics, and F type plasmid are specially for reproduction. Up to this we detail discuss. Here plasmid is a extra chromosomal DNA, additional molecule of circular DNA or mini chromosome, provide different functions. Up to this we detail discuss structure of prokaryotic cell. A review today's topic. We learn today's lecture kinds of cell. You know, all living organisms are grouped under two categories, prokaryote and eukaryote. On the basis of prokaryote and eukaryote, prokaryote are made by prokaryotic cell, eukaryotes are made by eukaryotic cell. So cells also two types, prokaryotic cell and eukaryotic cell. We learn in today's lecture prokaryotic cell in detail. What is actual prokaryotic cell, characteristics of prokaryotic cell, envelopes of prokaryotic cell, different cell organelles of prokaryotic cell. On the basis of today's lecture, some exercise question noted here. Number 1 MCQ question. What is nucleoid of circular DNA, double stranded DNA, both A and B, none of this? The correct answer is circular DNA is nucleoid. Resistant to antibiotics is conferred by plasmid, chromosome, nucleoid, both A and B. Yes, the correct answer is plasmid is provide resistant to antibiotic. Where in genetic material located in prokaryote? Nucleus, cytoplasm, ribosome, all of these. Yes, the correct answer, genetic material is located in prokaryotic cell or prokaryote, cytoplasm. So B, cytoplasm is correct option. What is external to cell wall in prokaryotic cell? Plasma membrane, cell wall, glycocalyx, none of this. Yes, the correct answer, glycocalyx is external to the cell wall. Means C, glycocalyx is correct option. Short answer question number 1. Function of glycocalyx, yes, adhesion to the bacterial cell or bacteria. Which type of ribosome present in bacterial cell? Yes, 70S type of ribosome present in bacteria. How prokaryotic cell differ from eukaryotic cell? Yes, prokaryotic cell have well organized nucleus is absent, while eukaryotic cell have well organized nucleus is present. So this is the difference between prokaryotic cell and eukaryotic cell. What are the envelopes seen in prokaryotic cell? Yes, glycocalyx is outermost envelope. Below the glycocalyx, cell wall is a second envelope, and below the cell wall, innermost envelope called as plasma membrane. What is plasmid? In prokaryotic cell, present one or two additional circular DNA, also called as mini chromosome, such structure called as plasmid. Which type of plasmid used for reproduction? Yes, F type of plasmid are useful for reproduction. Function of mesosome. Yes, formation of cell wall is one of the function of mesosome. Also, it helps replication of DNA. Which type of typical replication seen in prokaryotic nucleoid DNA? Yes, theta model of replication seen in prokaryotic nucleoid DNA. Which organ help rotatory movement of bacteria? Yes, flagella and cilia are provide rotatory movement of bacteria. Match the column A with column B. Glycocalyx function of glycocalyx, adhesion of bacterial cell wall. It gives mechanical support to the cell, means two match with A, here one match with C. Plasma membrane, yes, plasma membrane help intercellular transport, means 3 match with B. Identify the given diagram. Yes, this is the prokaryotic cell or bacterial cell. Labeled them A, B, C and D. Yes, here A is nucleoid or chromosome of prokaryotic cell. B, flagella of prokaryotic cell. C, pili of prokaryotic cell. And D, plasmid of prokaryotic cell. Up to this, today's lecture completed. Hope that you understood everything. If you like the video, then you can subscribe my YouTube bioinfo channel. Tap the bell icon and like the video. If any doubt about today's lecture, mention in comment box. Thank you.